Hi everyone, it's Dr. A, and in this video, we'll be investigating the first component of a process we refer to as cellular respiration, and the name of that process is glycolysis. Right before we begin, let's discuss the term glycolysis. One of the great things about anatomy or physiological concepts is that the name of something or a process gives us an indication as to what it does or what's taking place. And the same is true with the term glycolysis. If we break the term in two, the prefix glyco refers to glucose, and as you may already know, the term lysis means breakdown. So if we put those words back together again, glycolysis means glucose breakdown. Now, the question we should tackle is, why do we take the time to study or investigate this process? Well, the answer here is that it's a process that happens inside of you on a cellular level. But to give greater context, it's a part of the process in which your body generates ATP, which is the energy currency of the cell. And without ATP, our cells are unable to function. Now, let's take a moment to investigate the structure of the cellular membrane. At the cell membrane, we have two major components. First, we have the hydrophilic head, which is known as the water-loving portion, and then we have the hydrophobic tails, which is referred to as the water-fearing portion. And if we investigate these further, we'll see that within the hydrophilic head, we'll find a phosphate group and glycerol, and that the hydrophobic tails include one saturated fatty acid and one unsaturated fatty acid. Now, collectively, we call the cell membrane a phospholipid bilayer, and here's where we get this terminology. First, the prefix phospho comes from phosphate, and secondly, we use the term lipid to represent the fatty acid tails. But what we don't want to forget here is that linking these two is glycerol. Now, as you may already know, we can refer to the cell membrane as a semi-permeable membrane or a selectively permeable membrane. This means that some of the particles are able to diffuse through the cell without direct assistance, and that molecules with specific charges or that are larger in size need a special channel in order to enter the cell. So with the cellular membrane in mind, it's important to note that molecules must enter and or exit the cell as part of normal cellular physiology. And one of the things that can be helpful in understanding how these molecules interact with or pass through the cell membrane is to group them. The first category we'll investigate are lipid-soluble molecules. And lipid-soluble molecules can pass through the cell membrane with relative ease, unaided. Examples of these molecules include oxygen, carbon dioxide, and vitamins A, D, E, and K. Next are non-lipid soluble molecules such as glucose and sucrose. These items on their own cannot pass through the cellular membrane. They would instead bounce off of the cellular membrane, which is an indication that we'll need an alternative means for glucose to enter our cells. And lastly, we have specific non-lipid soluble molecules. This would include examples such as sodium, potassium, and chloride. In order for these molecules to enter the cell, they must pass through a specific protein membrane channel. Now, let's peer into the first part of glycolysis, which we refer to as the investment phase. So let's think about this practically for a moment. When we think about making an investment into something, we put up money into or for something in hopes that we get a surplus at a future date. And that's basically what's happening in the investment phase. Our body is investing its energy currency, ATP, in order to generate a larger amount of ATP at the end of this process. One of the first things that's important to make note of as we begin this process is to point out that the process of glycolysis occurs in the cytoplasm of the cell, and holistically, glucose is able to enter our cells because of the hormone insulin having its effect on the body. So now that we have its location in mind and what initiates this process, let's think back for just a moment. We can identify glucose by its chemical formula C6H12O6 as a non-lipid soluble molecule. And if you remember, we said that on its own, it's incapable of passing through the cellular membrane unaided. So because of this, it needs assistance to pass through. And the way that glucose does this is through a glucose transporter, which we'll refer to as GLUT for short. Now, it's helpful to note that these transporters are not the same as the protein channels that nonspecific lipid-soluble molecules travel through, although they do share some similarities. And here's something that's important to note about the GLUT channel. It allows glucose molecules to easily flow into and out of this passageway with relative ease. So, for a lack of a better phrase, if this molecule of glucose was left to its own devices, it could easily flow back out of the cell through the GLUT channel. Now, as we begin diagramming this process, we'll start by utilizing a combination of 6 green circles to represent each of the carbons we find in this molecule of glucose. So now that glucose has entered the cytoplasm, adenosine triphosphate, or ATP, arrives, and ATP donates one of its phosphate groups to the glucose molecule. And what this does is help to secure glucose within the cell so that it doesn't exit. Now, it's important to note that this chemical reaction takes place because of the presence of an enzyme, and this enzyme is either hexokinase and or glucokinase. So one of the things we should address while we're here is why it's both and or when one is more active than the other. Hexokinase is the more prevalent enzyme. It functions when glucose levels are low and helps in general to maintain a balanced level of blood glucose in the body. It's most prominently used in non-liver tissue, such as in the muscles, the brain, or fat cells due to their constant need of glucose for energy. On the other hand, glucokinase is the enzyme that becomes most prevalent when blood glucose levels are high in the body. For example, after we eat a meal that's comprised largely of carbohydrates, this enzyme becomes activated in the liver and or in the beta cells of the pancreas. Now, after the conclusion of this chemical reaction, we call it glucose 6-phosphate. And essentially, this means that we now have a phosphate molecule attached to the 6th carbon of our molecule of glucose. Now, our glucose 6-phosphate molecule goes through a process known as isomerization. And isomerization is basically a fancy way to say that we have the same molecular structure as before, but now the molecules have a different arrangement. So that we can see this with greater clarity, let's take a look at our glucose 6-phosphate molecule on the left and our fructose 6-phosphate molecule on the right. What we'll notice is that both are virtually the same structure. However, the difference is in the change in position of the carbonyl group shown on the first carbon molecule to now being on the second carbon molecule. And it's also helpful to note that this chemical reaction is facilitated by an enzyme called phosphoglucose isomerase, again giving us what we now call fructose 6-phosphate. Now we get a little bit of deja vu here because now our fructose 6-phosphate molecule undergoes phosphorylation, meaning that an additional phosphate group is added, and this occurs because of the presence, again, of ATP. Now, this chemical reaction is made possible because of the enzyme phosphofructokinase, and we now refer to our structure as fructose 1,6-bisphosphate. And the name of this molecule is a representation of the process that just occurred. This means that we have a fructose molecule that is now carrying 2 phosphate groups, one on the first and the other on the 6th carbon of our molecule. Now we have an enzyme called fructose bisphosphate aldolase, which acts on fructose 1,6-bisphosphate, splitting it into two separate molecules. Both of these molecules feature 3 carbons and a phosphate group, and we call this first molecule dihydroxyacetone phosphate, and our second molecule is glyceraldehyde 3-phosphate. Now, the second of our molecules, glyceraldehyde, is prepared to undergo the next stage in glycolysis. However, before we continue, our first molecule needs to be reconfigured so that we have 2 identical molecules moving forward. So, to become reconfigured, the enzyme triose phosphate isomerase isomerizes dihydroxyacetone phosphate so that it mirrors glyceraldehyde 3-phosphate, giving us a total of 2 of these molecules. Now, this represents the end of what we call the investment phase. And as a reminder, we call this the investment phase because the body has utilized or donated 2 molecules of ATP to this process with the intention that a larger number of ATP molecules will be produced. So here's a quick synopsis of the steps we've uncovered in the investment phase. First, glucose entered the cytoplasm through the glucose transporter. Secondly, ATP has donated a phosphate group to the glucose molecule, and we refer to this molecule now as glucose 6-phosphate. Third, glucose 6-phosphate now undergoes isomerization, which is essentially a reconfiguration of the molecule to become for us fructose 6-phosphate. Fourthly, another molecule of ATP donates a phosphate group to the molecule, and we refer to this structure now as fructose 1,6-bisphosphate. And in the last step, our fructose 1,6-bisphosphate is split into 2 smaller molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. And as we mentioned, eventually our dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate, giving us a total of 2 of these molecules. Now let's discuss the second part of glycolysis, which we refer to as the payoff phase. So again, if we think about this practically for a moment, the name of this phase makes sense. As noted earlier, when we make an investment, we do so in hopes that we get a surplus or a payoff at a later date. And it's in the second half of glycolysis that the payoff phase becomes actualized. Specifically, it's in this phase that our body begins accumulating a larger amount of ATP, which again is the energy currency of the cell. As we resume this process, we'll need to keep in mind that we have 2 glyceraldehyde 3-phosphate molecules. So in other words, what's diagrammed here occurs twice, but we'll focus on showcasing what happens with one of these molecules to explain how this process continues in the payoff phase. So with our glyceraldehyde 3-phosphate molecule, we have an enzyme acting on it called glyceraldehyde 3-phosphate dehydrogenase, which oxidizes the aldehyde group of the molecule. So to give greater context, here's what our molecule looks like, and here's the aldehyde group shown on it. In other words, it's through the process of oxidation that we have the loss or extraction of 2 electrons and 2 protons from the aldehyde group. What we'll see next is that 1 proton is released into the cytoplasm, while the pair of electrons and remaining proton adhere to a molecule called NAD, which stands for nicotinamide adenine dinucleotide, a product that's formed in and is already present in the cell as a result of normal cellular metabolic processes. And as a result of this, NAD becomes NADH. The next component of this step is the adhering of a freely floating phosphate molecule in the cytoplasm to the glyceraldehyde 3-phosphate. And for notation purposes, we've made the freely floating phosphate molecule a different color as a sign that ATP was not utilized in this reaction. We now call this molecule 1,3-Bisphosphoglycerate, signifying that we have a phosphate molecule at both the first and third carbon. Now, we'll revisit the NADH that's produced here later on, as it will be helpful in the generation of ATP. With our 1,3-bisphosphoglycerate, we have another enzyme acting on this molecule called phosphoglycerate kinase. With its presence, it facilitates the donation of a phosphate group to adenosine diphosphate, or ADP, which is already present within the cytoplasm. Of course, once this phosphate group joins with ADP, it forms ATP, or adenosine triphosphate, the energy currency of the cell. And what remains from this molecule is referred to as 3-phosphoglycerate. With our 3-phosphoglycerate molecule, we have an enzyme acting on it called phosphoglycerate mutase, which facilitates an isomerization, a process by which the phosphate group on the third carbon molecule moves to the second carbon of the molecule. With our 2-phosphoglycerate molecule, we have an enzyme acting on it called enolase, which leads to a dehydration reaction in which water is removed from the molecule, leading to the formation of what's called Phosphoenolpyruvate, or PEP for short. In essence, our PEP molecule still maintains its 3-carbon base with a single phosphate group. With our phosphoenolpyruvate, we'll have an enzyme that acts on it called pyruvate kinase, which facilitates the transfer of the phosphate group to an available molecule of ADP within the cell, making another molecule of ATP. This leaves behind for us a molecule we'll refer to as pyruvate. So this concludes our process of glycolysis. So let's take a moment to do a recap of the payoff phase. In our payoff phase, we began with glyceraldehyde 3-phosphate converting into 1,3-bisphosphoglycerate, and as a byproduct of this reaction, we have the creation of NADH, which will be utilized later on to generate additional ATP for the body. Next, we have our 1,3-bisphosphoglycerate molecule that donated a phosphate group to an existing molecule of ADP, making ATP and becoming 3-phosphoglycerate. Next, our 3-phosphoglycerate undergoes isomerization, which is essentially a reconfiguration of the molecule to become 2-phosphoglycerate. In the next step, our 2-phosphoglycerate releases water from the molecule, creating phosphoenolpyruvate, or PEP for short. And in the last step, our phosphoenolpyruvate transfers its phosphate group to a molecule of ADP, creating another molecule of ATP and leaving behind the molecule pyruvate. Now that we have our summary of the payoff phase, the next thing that we should consider are the end products of glycolysis. So in total, our bodies invested 2 molecules of ATP into this process, meaning that we get a yield of 4 ATP and 2 NADH, which equates to a net of 2 ATP, 2 NADH, and 2 pyruvate molecules. And last but certainly not least, it's important to note that glycolysis doesn't occur as a separate process. It continues, when special conditions are met, through what we can refer to as the citric acid cycle, or Krebs cycle.